Good morning, good afternoon, good evening. Welcome back to chapter 15. We just got done talking about the early Earth. We just got done talking about how we date fossils, how we find out how old fossils are. And this is kind of fundamental to our understanding of how old the Earth is. As we look at how old the rocks are, we use that radiometric dating to figure out the age of the planet. First life probably didn't start until about 1,000,000,000 years into the age of the planet, and 1,000,000,000 years, we use that number, we throw it around, but I want you to understand what it really means, 1,000,000,000. 1,000,000,000 means 1,000,000,000. If I had a calendar that kept track of 1,000,000 years, I'd have 1,000 of those calendars. That's what 1,000,000,000 means. 1,000,000,000 is a really long time. How did we go from a lifeless hunk of spinning rock to this life-covered hunk of spinning rock that we see today? Life probably originated over over 4 different steps. The first step is the abiotic creation of organic compounds. Abiotic. What does abiotic mean? Well, bio means life, and what does it mean when you put a or an in front of anything? It means without. So abiotic means without life. So we're talking about the creation of life chemicals from non-living sources. Examples of these organic chemicals would be like some simple amino acids, some nitrogen bases like the A's and the T's and the G's and the C's that we find in DNA. We can actually make these things from non-living sources. Living things are really good at making them because it's essential to life. But non-living processes can accidentally make these things. Amino acids will form peptide bonds when soups containing amino acids are allowed to dry on hot rocks. So we've got that early Earth atmosphere, which has a carbon source, it's got a nitrogen source, it's got a hydrogen source. The electricity from lightning bolts, the heat from volcanic eruptions, the hot rocks from volcanic eruptions, all of these things together, energy source plus precursors, chemical precursors, plus a really long time, like 1,000,000,000 years, can lead to accidentally creating this thing that we call life. Carbons plus nitrogens plus hydrogens. You mix those together and we get these things called amino acids. When amino acids are allowed, when liquid, like, the ocean, when soups containing amino acids are allowed to dry on hot rocks, they'll spontaneously form those peptide bonds. The peptide bonds, which you string enough amino acids together, at least 50 of them, and what do you get? You get proteins. So again, allow this to happen for a few 100,000,000 years, and you can get the first stages, the first things that we need to get towards life. After we can make proteins, now we need to package these proteins. Now we need to package the molecules of life. And these packages of these organic molecules we'll call protocells. Proto means beginning or early. Cell, obviously, is the cell, so beginning or early cells. We talked quite a lot about phospholipids. We know that phospholipids, the balloons, the two squiggly tails, phospholipids, when you have a collection of them and you force them to deal with water, they will spontaneously form a ball like this, with all of the heads on the outside and all the tails in the center. Take that thought a while farther and we can finally get to making very simple phospholipid bilayers and get very simple cell membranes. Those cell membranes, some of them accidentally can contain proteins and other organic molecules. Now that I've contained these organic molecules, I can now do one of the things that life requires, and that is that life has its insides separate from its outsides. Remember that these phospholipid bilayers, these simple membranes, are very difficult for organic molecules to get through. They can't get through because of those lipid tails in the center. So inside, we've got basically an isolated seed. We have an isolated soup where now we can change. Now we can do some special chemical reactions that require molecules to be concentrated. If it's all just floating out everywhere, it's really difficult for the molecules to get together. But now if I've got this membrane, now if I've got stuff enclosed, those chemical reactions can take place a lot easier because they're within an enclosed space. They can't just drift away from each other. First, I've got to make things like proteins and simple nucleic acids. Second, I have to enclose those proteins and simple nucleic acids. Third, those simple nucleic acids can self-replicate. RNA can replicate itself. All you need is just the raw materials, those A's and U's and G's and C's. That's one of the really cool things about RNA is you give it the raw materials and it will just copy the other side. Remember, if there's an A over here, what has to be on that side? It has to link to a U. And if there's a G on this side, it has to link to a C. You can't mess it up. So you get a faithful copy each time. Well, now we've got something that can replicate on its own if it's given the raw materials. We have an enclosed space where we can keep those raw materials in the same space as the parent chain. And we've got some proteins which could be used for a surface for these things to replicate on. These proteins can be in the membrane to allow only some things to come into my early cell and allow other things or keep other things out. Self-replicating molecules like RNA. DNA is not as self-replicating as RNA is. RNA was probably the first genetic code of life. DNA probably came later. Remember, if I've got the monomers, if I've got the separate pieces that go into making RNA, A's and U's and G's and C's, they will spontaneously make chains like this. And then now that I made a chain like that, I can make the other side. And once I can make the other side, I can keep replicating back and forth, back and forth. If I've got one side, I can just make the other side. This self-replicating ability that RNA has makes it so that you can have instructions, and those instructions can be faithfully copied, those instructions can be faithfully followed. Stanley Miller did the first experiments on making biological compounds. He took some of those compounds that we thought may have been in the early Earth's atmosphere, like methane, like ammonia, like hydrogen, and he mixed them in what was called a reaction chamber. It's a lot more complicated than this, but a reaction chamber is essentially a sealed test tube, a big sealed beaker, that he then added sparks to. I got a picture showing you what it looks like in just a minute, but at the moment, it's a contained system. It's a sealed system with ammonia gas, with methane gas, with water vapor, with hydrogen. So these gases floating around in there. He basically puts a scientific spark plug in it and then added sparks to this gaseous mixture. Notice there's no oxygen in there, so no explosions. He let that run for a few weeks, and he collected out of the bottom of it the liquid that was formed. And then he ran that liquid through some tests, and he found that he actually made simple organic molecules. Simple organic molecules like amino acids, simple organic molecules like some of the precursors to RNA, simple organic molecules like urea. Urea is that stuff that is in your pee. Urea is what makes urine smell like urine. But that is an organic compound. That is something that only life makes. Well, it can be made accidentally by non-living processes. Here's that reaction chamber I was telling you about. And again, it's a lot more complicated than just a sealed beaker, but it's a sealed system. In the sealed system, he has water. This C, this water contains methane. This water, this vapor, it contains methane and ammonia, hydrogen. The Bunsen burner is the heat source, as in like lava flowing into the ocean to make, hot water. The vapor escapes. It comes in here with the mixture of the methane, the ammonia, the hydrogen. We've got the scientific spark plug that's creating the electric current. And then it goes through a cooling chamber. The cooling chamber makes this a big circle like this. So the gas goes up, reacts, comes down, back and forth, back and forth, back and forth. It's a big sealed system. He can pull this little plug out and allow some of the liquid to come out, and then this is what he could test. And again, he let this run for a few weeks, and after a few weeks of letting that run, what he found is those simple organic molecules, some of the precursors to life.